start now, Tanya.
The joy of life comes from our encounters with new experiences, and hence, there is no greater joy than to have an endlessly changing horizon, for each new day to have a new and different sun. Don't settle down and sit in one place. Move around, be nomadic, and make each day a new horizon. On this note, I, Tanya Saxena, on behalf of Department of Botany, Deshbandhu College, would like to welcome our esteemed chief guest, Professor Renu Deswal, who has agreed to grace us with her presence. I would also like to welcome Honorable Principal, Professor Rajiv Agarwal, Vice Principal, Professor Kamal Kumar Gupta, IQAC Coordinator, Dr. Aditya Saxena, teachers, and all the students to the inaugural function of the Botanical Society of Deshbandhu College for the session 2021-22. Due to the pandemic, we are organizing this event digitally. We apologize in advance for any technical glitches that might happen. The Botanical Society, through its various activities, provides the students with an excellent platform to showcase their talents and skills. The various opportunities provided by the Society help in the growth of the students. The Society organizes various talks, excursions, and webinars by eminent speakers to encourage and enlighten the students. Every year, the Botanical Society comes together to put together its annual fest called Sanjeevani, which comprises of riveting competitions. Over the years, we have seen students from many colleges take part in the events and enjoy them. The work of the Society done during a year culminates with its annual magazine, Petals. It includes articles and art pieces submitted by the students and teachers alike. For today's inauguration event, we have organized a talk by distinguished and illustrious Professor Renu Deswal on the topic Proteomics to Nanoproteomics. We have also organized two events after this program, an online quiz competition called Lockdown Memories and a pot making competition called Rang De Matka. Saraswati Namastubhyam Varde Kamrupini Vidyarambham Karishyami Siddhir Bhavatu Me Sada. As is the tradition, we will start our program by lighting the lamp and invoking the blessings of the Almighty. Goddess Saraswati is an embodiment of wisdom, knowledge, and learning. We seek her blessings in all spheres of life. Let us join our hands in prayer as we light the lamp and listen to this beautiful rendition of Saraswati Vandana.
या कुन्देन्दु तुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा शुक्लां ब्रह्मविचारसारपरमा आद्यां जगद्व्यापिनीं वीणापुस्तकधारिणीम् अभयदां जाड्यान्धकारापहां हस्ते स्फाटिकमालिकां विदधतीं पद्मासने संस्थितां वन्दे तां परमेश्वरीं भगवतीं बुद्धिप्रदां शारदाम्।
A good teacher can inspire hope, ignite the imagination, and instill a love of learning. With these words, I would like to invite the teacher in charge of Department of Botany, Dr. Aparna Nautiyar, to say a few words.
Thank you so much, Tanya, for the kind words. Namaste and a good afternoon to all of you. On behalf of Department of Botany, Deshbandhu College, I, Aparna Nautiyal, welcome you all in the inauguration ceremony of Botanical Society today. First of all, I would like to extend a hearty welcome and express my sincere gratitude to our eminent speaker, a hard-working researcher and a dedicated professor, Professor Renu Deswal from Department of Botany, Delhi University, to give her consent and spare her valuable time for the inauguration. Thank you so much, ma'am. I would also express my gratitude towards our dynamic, hardworking, and dedicated leader of our institution, Professor Rajiv Agarwal, for his constant support and guidance in all the progressive activities of the department as well as college. He would have loved to come today, but because of his prior commitments, he has sent his wishes and blessings to all the students as well as the society. I also welcome and express my gratitude to our equally dynamic and supportive Vice Principal, Sir, Professor Kamal Kumar Gupta, for motivating all of us. Thank you so much, sir, for coming. Thanks are also due to IQAC convener, Dr. Aditya Saxena, for all his support and guidance. Botanical Society gives a platform to showcase the talent of the students apart from their all academic activities. I am also very much thankful and express my gratitude towards the senior colleagues of the department as well as our late Dr. Dharmendra Kumar Malik sir for his contribution towards the progress of the department as well as to the society. Thanks. I would also like to congratulate and wish all the best to our highly energetic and charming society advisor, Dr. Sunita Malik, and all the student council team for all the future activities throughout this session. So I welcome you all in this inauguration function. And I am very much hopeful that the talk by Professor Renu Deswal would be highly insightful and fruitful to every one of us. So thank you so much for participating today for this inaugural event. Over to you, Tanya.
Thank you, ma'am, for your words of wisdom. Now, I would like to invite our respected Vice Principal, Professor Kamal Kumar Gupta, to enlighten us with his words.
Yeah. Good afternoon. Am I audible? Do you hear me?
Yes, sir.
Yes, sir.
Yes, sir.
And a very warm good afternoon, a bright sunny afternoon on this spring season. I welcome you all for the inaugural session of Botany Department, Botanical Society 21-22. First of all, I'd like to welcome on behalf of Deshbandhu College and on my own behalf, today's speaker, Professor Renu Deswal, Professor in Department of Botany, University of Delhi. Yep. Welcome you, ma'am, and thank you for accepting invitation for the inaugural talk. I welcome our dynamic, visionary, versatile, respected principal, Professor Rajiv Agarwal, who is always a constant support for all the events organized in Deshbandhu College. I can see many of the senior faculty of Botany Department in these events, especially Dr. Veena Ganju, I can say. Welcome, ma'am. And you are always a support for all the events. I welcome senior faculty members of Botanical Department, especially Dr. Sarla, Dr. Bela Bhatia, Dr. Roshni Rajamohan, Dr. Kumar Shantanu, Dr. Rajender, and a very dynamic and versatile teacher in charge, Dr. Aparna Nautiyal. Welcome you all. My welcome to all the faculty members of Deshbandhu College and faculty from other colleges. Welcome all of you. Then I congratulate Botany Department for taking lead despite of the adverse conditions of pandemic, and for that the wind beneath the wings of Botanical Society, Dr. Sunita Malik. Congratulate Dr. Sunita for organizing such a wonderful and taking initiative despite all adversities of the pandemic. Congratulations.
Thank you, sir.
Congratulations and best wishes to the Botanical Society, and especially the organizers, the team members, Priyanka, Chaitanya, Neha, and Tanya. My best wishes and congratulations. Now, related to the topic, I being from the same subject or related subject, the adjacent is from zoology department. The talk of today's talk, proteomics to nanoproteomics. But madam, let me say, we have seen an era from genomics to proteomics to omics and then nanotechnology. So to understand what nanoproteomics would be there, it is so recent and it has got a tag, yeah, that is one of the finest study. I was just going through Google and they say a marriage of nanotechnology and proteomics, and that can really bring wonders. I've seen a time I was working on some of the nano formulations for my research work, but we never thought that nanoproteomics can also be there. Nanoproteomics, they say that it is the proteome analysis of non-abundant protein. And especially when you have very few cells in the culture and you want to go for the proteome analysis of that. So that can really bring wonders in the future. And of course, with that very little knowledge, we will be really privileged, and our understanding about the subject would be great by this talk of our respected professor in this world. So without taking much of the time, I think all of us are eager to hear the talk. So I think over to you, Tanya. Thank you.
Thank you, sir, for encouraging us with your words. Moving ahead, I would like to invite Ms. Priyanka Barua from BSc Honours Botany 3rd year to take over and introduce our chief guest for the day.
Thank you, Tanya. It's well said. There is always light. If only we are brave enough to see it, if only we are brave enough to be it. It's my distinct honor to introduce to you our esteemed guest speaker for today's webinar, Professor Renu Deswal. Her name is renowned globally in the field of botany, and she is a person with an essence of service, dedication, and compassion. Allow me to introduce her career, work, achievements, and professional roles. Professor Deswal did her graduation from University of Delhi, followed by MSc, MPhil, and PhD from Jawaharlal Nehru University. Her areas of specialization include abiotic stress, functional genomics, nitric oxide signaling, and nanobiotechnology. Professor Deswal began her career as a research associate and then as a research scientist at JNU. She then served as a staff scientist at the National Center of Plant Genome Research at JNU from 1998 till 2001. She joined the Department of Botany at University of Delhi as a reader in 2001 and became associate professor in 2006. In 2009, she became professor and still continues to deliver her service to the department. Professor Deswal has successfully supervised more than 9 doctoral theses and 15 MPhil dissertations. She has also been a part of major 2 projects funded by DST PERS and DBT IBSD respectively. She has, to her credits, more than 85 publications in the Journal of National and International Repute, including research papers, book chapters, and popular articles. She has many prestigious awards and distinctions under her name, including the Springer PSI Award for holding the second highest number of publications in the journals of proteins and proteomics in the past 10 years, presented at the annual meeting of the Botanical Society. She has also been awarded with the National Award for Significant Contribution in sea buckthorn research by the Sea Buckthorn Association of India at the 7th Conference of the International Sea Buckthorn Association, among many others. Professor Deswal is associated with many professional bodies, including Executive Committee member at the Sea Buckthorn Association of India, invited member for New York Academy of Science in USA, life member in Third World Organization for Women in Science, Italy, and so on. She has served as an editor with prestigious journals such as Frontiers in Plant Physiology and as co-editor with Professor Sanjeev Srivastav and Suman Kundu for a special volume of journals of proteins and proteomics known as Functional and Interaction Proteomics, Application in Food and Health, among others. She has been a significant part of various administrative committees. She has been appointed as the chairperson managing committee for Rajiv Gandhi Hostel for Girls, University of Delhi, chairperson of governing body in Atmaram Sanatan Dharma College and Acharya Narendra Dev College, New Delhi, deputy proctor in North Campus, University of Delhi, member board of studies at University of Haryana, Mahendragarh, and MDU Rohtak, and many more. Professor Deswal is not only a distinguished researcher, but also a great teacher as well. Currently, she is engaged in teaching papers on nanobiotechnology to MPhil and PhD students, and paper on physiology and biochemistry, genomics and proteomics, topic in plant physiology and biochemistry to MSc students at Department of Botany, University of Delhi. You're truly an inspiration, ma'am. Now, I would like to invite our speaker, Professor Renu Deswal, to grace the dais and enlighten us with her talk. We welcome you, ma'am.
Thanks, Priyanka, for your extensive and very nice introduction. It's my pleasure to be here, and the pleasure is actually even more when you are delivering your lectures to the students, because if you can somehow excite them, if you can generate some interest in them, that is the main aim of presentation. And I have actually kept my presentation a little simple, keeping the students in mind so that I can deliver some basics to them. And before starting my presentation, I would like to formally thank Professor Agarwal, Professor Gupta. Professor Agarwal is not there today, it seems. So Professor Agarwal, again, for the kind introduction of the area. And that is the area, that's the actual thing, the marriage of nanotechnology and proteomics, that is nanoproteomics, as you mentioned. So I'll be touching on to that for benefit of students, the areas, introduction of those, and then how this area develops and what little we have initiated in this area, and a small piece of work. And I would also like to thank the teacher in charge, Aparna ji, Dr. Aparna, for your kind words. And she's our student. She's from the department. So you always have a special bonding with your students. You always, whenever you see them, you feel so excited and you feel so nice. And Sunita has been in our lab for some time. So I know Sunita also in that sense, and all senior teachers who are present today of botany department and of other departments. It's a great thing to be here, and my regards and my hello to everyone. And I think I can start with my lecture now. And there are a few more students. I think there's one more student. I'm forgetting the name now. A boy who is from the department in Deshbandhu. अभी नाम याद नहीं आ रहा है, याद आएगा तो फिर बताएंगे. a tall boy? Shantanu, yeah, Shantanu is there in the college. So anyway, I think I can start my lecture now, and I'll just share my slides and I'll start with the presentation. Just a minute, I'll share the screen and just tell me if you can see the screen properly. I'll just go to the slide presentation mode. Is the slide visible?
Yes, ma'am.
Okay. So the topic of the presentation, as Dr. Gupta mentioned, it's proteomics to nanoproteomics. And so keeping the topic in mind, what I have done, I would try to introduce both areas a little bit, and then I'll come to nanoproteomics in the end. So few slides on proteomics for the benefit of the student. I know all teachers, they must be aware of this now. These are routine slides, but for the benefit of the students, I thought I'll give some basics and then I'll move ahead. Then some tools and techniques of proteomics, which we use generally to do these analysis. Then some on applications, what information can you dig out, some limitations, then introduction to nanotechnology, and lastly, nanoproteomics. Some interesting work we are doing with the protein corona. I'll also share what protein corona is. So that is very exciting piece of work, and we have just initiated it 3-4 years ago. So what is protein? Proteins are actually the executor molecules in any biological system, whether it is animals or plants. They are the doers. They do all the job. And generally, proteins have huge chemical and structural diversity, and that's how they can perform diverse functions in biological systems. In your system, you have structural proteins, like you have skin, muscle, hair, bones. All these are, they are constituted by structural proteins. But you have all the functions in the body, whether when you talk of digestion of food, you talk of nowadays immunology or immune system is very talked about because of corona, your immune system. So even your immune system, there are many proteins, basically your antibodies, the glycoproteins. And I'm sure you are very much aware about these in today's time. These antibodies, they are glycoproteins which are present, and they are the one who are actually taking care of our immune reactions. So when you talk of proteins, proteins actually, as I mentioned, they are the functional or the executors of the cell, executor molecules, and then they are very different. They are very, they have different types of proteins are there. And this picture is clearly shows here that this is an adult butterfly and this is the pupa, or both are two different forms of the same organism, but they are different because of presence of different proteins in them. The protein component or composition would be very different in the two. And these proteins, they are made up of the building blocks. And again, this is textbook knowledge. The building blocks are your amino acids. They form your primary structure. They form chains of proteins or polypeptides, which you call them. And then they can form a slightly higher organization as secondary structure. Finally, as tertiary structure, when more than one secondary structures, they join together, they form the tertiary structure. And finally, the final shape or final protein, the quaternary structure is produced, which is your functional or the main protein. You must have also heard about certain damaged protein or misfolded proteins, which are leading to many diseases. Now we know more than 100 diseases, which basically they are affecting your brain functions, and Alzheimer is one of them. In animals also, we have mad cow diseases. So these are of serious concern. So protein, normal functioning of protein is very important. If you have any problem in function or structure of the protein, it would lead to diseases and even death if the things are very serious or very critical. So functioning, proper structure and proper function of protein is to be maintained for proper physiology, whether again in any living organism. Now, this is again, it is now what is proteome? This definition, the term proteomics or proteome, these were coined by this person, Dr. Mark Wilkinson. He is in University of New South Wales, Sydney, Australia. So sometime in mid 19, I think he coined this term. So proteome, basically it refers to entire protein complement of any organism. Say, for example, yeast has about 6,000 different proteins, and in human we have actually these are the number of genes. Protein number is quite high. In humans we have about 32,000 proteins. In Arabidopsis about 25,000. So these proteins we can deduce their function from their sequences and their structures, and also from analyzing their interactions with other protein and mutating them and knowing about the function and even understanding the complexes in which they are involved, and that is how you can guess their function. So there are various ways by which you can analyze functions of protein. I'll go to the next slide. So now what is proteome? So as I mentioned, entire protein complement of an organism is known as proteome. So now as Dr. Gupta was mentioning that now we have many omics areas of research which has come into existence, or actually they are necessity, and why we need them, and what is the reason why there's a need of these omics areas of research. So when you talk of proteomics, normally this is your central dogma. From your DNA, by transcription, you get formation of messenger RNA, and then that messenger RNA is translated. It is transported to your cytosol, and then it is translated to proteins. And these proteins in turn can be then modified by PTMs, or even other modifications happen. So if we analyze transcriptome or transcripts, this information shown in this circle, this is what we always miss on. So this is where this gap is filled by proteomic analysis. So this is a diagrammatic representation of the same information, because precisely one gene is no longer equal to one protein, or one gene does not code for one protein. It can have, one gene can code for multiple proteins. Now, to know about those multiple proteins, you need analysis at proteome level. Okay, this is the same information. Transcription gives rise to RNA, then after processing, this leads to your messenger RNA, which is translated, and these are the modifications and which happen later on, either proteolysis or PTMs, post-translation modifications. So our proteomic analysis helps in understanding these things. And another reason for this is these genome projects, which have happened in last, say, 3-4 decades, they have created sea of information. And we have to make sense of this information. What is this sequencing data, and what is the meaning of this? So this is where then again a branch of proteomics called functional proteomics helps us to know what these gene sequences are coding for. And one important point, generally when you sequence a genome, you have to annotate it. You have to know what portions of genome are coding, what are non-coding. You want to understand the whole sequences, what they are, what they mean. So once this kind of analysis is done, then this is one important piece of information which has come forward, that almost 2/3 of all the genes which are identified, their function is not known. So important point which we get from this information is that we know lesser than what we don't know. We just know functions of 1/3 of the genes. 2/3 we don't know. So this is where, because we don't know the variants. So this is where your proteomic analysis comes handy, and it helps you to understand these unknown functions of the coding genes. So proteomics, as I mentioned earlier, it's the study of proteome, and it's basically your full complement of protein in organism, or if it's a single cellular, it will be the single cell, or if it's a multicellular, it can be at different levels, organelle level or at tissue level and all. So the classical definition of proteomics is basically large scale analysis of proteins. Now we don't just, this is not enough actually. We have to now make sense of this information also. So we take help of other areas, like we take information from transcriptomics, then genomics, then we also try to understand the interaction between protein just to know their function. And this is an important point about proteome, the third point, which is mentioned in the slide, that the proteome is dynamic. Dynamic means it is always changing. It is not static. Like mostly our genome is said to be static. The changes in genome are very slow. Mutations happens in years and years, but proteome is changing in spatial form also. That means location wise also, and temporally with time also. So they are both kind of regulations which change your protein. Excuse me. So understanding the proteome allows us to characterize protein, to understand protein interactions, and identification of biomarker is also possible by doing proteome analysis. And this is a simple workflow which has been shown how you actually go for identifying your proteome. There are various branches or various possibility which you can follow, but generally the workflow is like this. You have to first extract your protein mixture from whatever source you are trying to analyze your proteome. Then you segregate those proteins. So there are separation methods, either gel-based or liquid chromatography-based, and you segregate these proteins, and then you digest them. Why we digest them? We digest them because our mass spectrometry machines, they can identify peptides better. Okay, if you have longer proteins, then they generally errors crop in. So generally for MS analysis, mass spectrometry analysis, you have to digest your protein with, and the most commonly used protease which is used for digestion is trypsin. Okay, so you trypsinize your proteins and then you feed them into MS machines. I'll be using the word MS for mass spectrometry, and then you can, there are ways by which you can identify those. Okay, so these are the major methods or steps when you are doing proteomics. So as I mentioned, first you have to make extract, then you separate your proteins, and separation is done to just to reduce the complexity of your protein sample. If it is very complex, then the spectrum which you get out of your MS machine would be very difficult to comprehend. So that's why you have to simplify or extract, or you have to purify it to some extent. That's why separation step is very important. So you separate them on gels. You can also use certain immunochemistry techniques to kind of do segregations of your protein. And chromatography also is used many a times to segregate or separate your protein mixture. Then, as I mentioned earlier, identification is done by mass spectrometry majorly, but you take help of immunochemical steps also sometimes to just to understand. Then finally, once you have identified your protein by mass spectrometry, and when we are identifying them, actually quantitation is also possible. Depending on the requirement of your experiment and what actually you want to derive from experiment, you can choose for the mass spectrometry analysis, whether you just want to identify protein or you also want to quantify them. For that, there will be two different ways of doing that. So the selection of the equipment also would depend on that, what kind of information you want to derive. And then eventually you do a lot of database search to make sense of whatever information you have obtained from your mass spectrometry identification. And this is again just for the students. I am sure they must be aware of this SDS page. It's a cornerstone technique for proteomics when you're doing gel-based proteomics. So, but this technique, this SDS page, or sodium dodecyl sulfate polyacrylamide gel electrophoresis, the full form of the procedure is this. This was this person, this scientist, Arne Celius, he has contributed a lot to this, and that's why he was, he's called as father of electrophoresis, and he was conferred a Nobel Prize. And Nobel Prize, again, I'm sure you are aware that these are given to excellent work which is done by a scientist in any area of science, and there are different Nobel Prizes which are conferred. So he was conferred Nobel Prize for his contribution to electrophoresis. And this is the basic principle of SDS page. You simply doing the separation on basis of variation in size of proteins, and you use basically an anionic detergent, SDS. That is how the name of the technique, SDS page. and you just label your proteins with this, you just mix SDS, and so that they acquire a net negative charge, and then you separate them because the charge is uniform, so the separation is on basis of size only. And in this picture on the side, you see a stained gel. After separation, the gel is stained with the Kumasi brilliant blue. So you can see blue polypeptides on gel. So this is one very commonly used technique for gel-based proteome analysis of proteomes. Then this is how your 2D gels look like. They would show as spots. And the only difference between SDS page and your 2D is here another property of proteins, the charge on protein is exploited. So first, in first dimension, you separate your protein on basis of charge, and then you lay out this strip on top of SDS page, where the principle again remains the same on basis of size. And eventually the result you get in form of spots in the gel. And these is the apparatus. This is your first IEF running machine where you separate or segregate your protein on basis of charge. And this is your routine apparatus, the second one, SDS page gel tank. So, and as far as chromatography is concerned, there are basically three types of chromatography, which are generally very commonly used. And again, this this Russian fellow, Mikhail Semnovich, he is considered as father of chromatography. So the three types are the separation when you are using matrices for separation on basis of size. You can also separate on basis of charge, and you can also separate on basis of affinity of molecules. This is called affinity. That is why it's called affinity chromatography. So you can hook on an inhibitor or a drug or any molecule which can interact with your protein of interest. They are kind of hooked to the column matrix, and then you can separate your protein of interest. So this is another way of segregating or separating your proteins before going ahead for their identification by mass spectrometry. And in mass spectrometry, basically, the principle of identification is on differences in masses of molecules. Okay, and using mass spectrometry, you can identify molecules which are present in solids, liquids, or gases, and you can even know the quantity of each type of molecule and their structures also, what kind of interactions they are having, what kind of bonds they are having. So again, these are the people who have contributed to mass spectrometry: John Fan, then K. Tanaka, and Kut. These are the three people who have again awarded Nobel Prize in Chemistry for their contribution to development of mass spectrometry. It is something now which is being used for knowing structures, knowing proteins, identifying proteins in case of animal and plant systems. In chemistry, they are being used. Definitely they are high-end machines, but mass spectrometry is being used to know even the structure of molecules and biochemistry people. So generally in a mass spectrometer, there are three things which happen. The important condition is your sample is to be in ionized state. So you do ionize your samples, then there's a mass chamber, or again the segregation of these ionized species happens, and then lastly detection is done. So eventually the output of a mass spectrometer analysis is a mass spectrum, and that is what you get. And then the next steps are basically database based, and you use several softwares to dig out the information from your mass spectrum, what this mass spectrum means. So the mass spectrum basically tells you about the protein being identified. So it will give you, once you basically do matching, you do matching of your mass spectrum with the theoretical information, and that is how you go about identifying your proteins. And this is one of such methods. It's called peptide mass fingerprinting, or PMF in short. Again, the three people who have contributed this is Hangel, Watanabe, and John Stutz. So these are the people who contributed a lot to development of this procedure, peptide mass fingerprinting, by which you can identify protein using a simple mass spectrometer Malditov kind of machine. So you can identify your protein after processing them through the steps I've told you. And what are the benefits of proteomics? It can tell you about splice variants. It can tell you about post-translation modifications. And phosphoproteomics is one area which has been worked on a lot in comparison to other modifications. But other modifications are also being analyzed, hundreds of them. And just to tell you that we know more than 400 modifications, protein modifications, which are possible So there's a lot of information which needs to be dig out, but a lot of work is already being done. And you can also know about biomarkers. Biomarkers basically would be proteins which would be linked to a particular stage. It could be normal growth and development, or it could be a disease-linked biomarker. So those kind of analysis are also possible by doing proteome analysis. And again, continuing the same, why we are doing proteomics, because it helps us in functional annotation of genome. Normally, when genome annotation is being done, as I mentioned, that we just know about 1/3 of the proteins, their function. We don't know about 2/3 of them. So this is where your proteomes come handy, or proteome analysis comes handy. And we have a branch of proteomics called proteogenomics, which has helped us to correct our drafting or annotation of genomes. And there are many corrections which are being done, where like kind of some regions which were earlier predicted as non-coding, now they are appearing as coding after these kind of analysis. So those corrections are possible. Then, as I mentioned earlier, protein function, protein post-translation modification, then localization and compartmentalization you can know by knowing the single peptides which are sitting on the protein. Similarly, protein-protein interactions and protein differential protein expression studies can be done by a proteome analysis. This is all possible. And there are three basically different types of proteomics broadly. Protein expression studies, as I mentioned, differential protein expression studies can be done. Then structural proteomics basically deals with the 3D structures of protein, or information about the crystallized protein or their structuring as such. So either X-ray crystallography or NMR studies, they basically enrich this area of research. And by knowing structure, you can again predict the function. You can know about the function of protein. So this is another way functions of protein can be known. Then even interactome analysis is being done. That also is contributed to functional proteomics in a big way. And this functional proteomics information is in turn helping us to annotate our know the functions of genes. As I was mentioning, we don't know the functions of 2/3 of genome sequences. So this is where all this information is helping us to understand the functions of proteins. And this is just a summary slide about proteomics. I've already mentioned most of these things, so I'll just skip this slide because parts and bits of this I've already discussed. So now we go to the last slide on, I think this is the last one on challenges of proteomics. One big question is about native conformation of protein, because whatever I've mentioned till now, we are using SDS page. We are denaturing our protein and then we are cutting them down with our trypsin. So that means we are missing on the information which would be there because of the native conformation of protein, because we are handling the denatured protein or the sliced proteins. Okay, so this is one big question which is still there. Like whatever information we are getting, although we are getting a lot of information, I won't say that we are not getting information, that information is not correct, but the information which resides in the native conformation, we are still missing on that. Then another limitation is like when we are doing our transcriptome or nucleic acid analysis, the speed with which we analyze protein is not, it does not match either the scale or the speed or the sensitivity, because as I mentioned, proteins are diverse in nature. So like we have probes for nucleic acids, we don't have similar probes in the sense the only probes available for studying proteins is antibodies. And the limitation is there because each protein has its specific antibody. You cannot have one antibody and that detecting many protein or identifying many proteins for you. So that is, probe is a big limitation. Another limitation which is related is we don't have protein PCR like we have PCR for nucleic acids. We can amplify our nucleic acids. Proteins, if they're low in amount, the only procedure is to purify them. That is the only way. Okay. Or you can at least put them in vectors or expression vectors, and that is how you can scale them up. But a PCR, protein PCR is not existing. So that is something which needs to be discovered. Then this I've already mentioned, we have specific antibodies, the last point. For proteins, we don't have probes which can detect a class of protein. Okay. Now coming to, I think, I don't know whether you can see the title of the slide. I think it is about nanobiotechnology. Somehow this thing is not showing it. But anyway, it is basically the analysis or the study of controlling of matter on an atomic or molecular scale. And the scale is in nanometer. When the material, whatever material you are analyzing or using for your work, that is in that dimension 1 to 100 nanomolar, that the nanometer, that is called as a nanomaterial. And these are the definitions which have been given for nanoscience and nanotechnology. Nanoscience is the study of phenomena and manipulation of material at atomic, molecular, and macromolecular scale, where properties differ significantly from those at larger scale. Nanotechnology is basically the usage of these material, the design, characterization, production, and application of these material or devices or systems, which are, again, the condition is they are at nanometer scale. Okay. So this is just a picture to show you the sizes of, so that you can just relate. Sizes of a water molecule is 0.1 nanometer. A glucose molecule is 1 nanometer. And a bacteria about 1000 nanometer, and this is the size of a tennis ball, just to give a relative relationship between these scales. And the important point when you're working at nanoscale, actually the properties change, whether it's your melting point or it's your boiling point or electrical conductivity or chemical reactivity of the material. It is actually enhanced because the surface area of this material is tremendously high. So even their physical properties, which are usual in normal material, they change. Okay, and there are various factors which would regulate this change. Or there's another term which is commonly, very commonly used when you are handling nanomaterial, that these nanomaterial properties can be tuned. Tuned means they can be changed or adjusted. And there are many factors which kind of control these properties of nanoparticle, like their structure, their shape, their size, and even their surface properties. They are responsible for their different behavior than the normal matter at nanoscale. And these nanomaterial, they can be either synthesized using chemical methods, or they can be synthesized using green methods, or when you're using actually plant extracts, that is called green synthesis, or for that matter, even fungal and other, that also comes under that. Basically, you're using material of biological origin for the reductive capacities. But in case of chemical synthesis, you use chemicals to make these nanoparticles. So definitely this green synthesis has several pluses, energy wise and usage wise, because you are not kind of leaving toxic material in the atmosphere. So this is a preferred way of green synthesis is a preferred way of making nanomaterial or nanoparticles. In nature also, nanoparticles are being made. And where are they being made? They are being made at site of fire, say a forest fire, or even in factories and all, industry and all, where a lot of heating happens. And even while cooking, they say that nanoparticles are produced in volcanoes. And our minerals deposit sites, those are the other areas where nanoparticles are kind of produced naturally. Besides this, as I mentioned, we can synthesize them, that is called engineer nanomaterial, and this is naturally made. So, and these nanoparticles or nanomaterial, they have application, they have entered our life like anything. You must have heard about their daily advertisement which comes on television regarding paints, like paints which on which dust does not stick, because these are specially designed paints with nanomaterial where the sticking of, like, they are slippery in nature. Okay, then you have even fabric now, which is available with antibacterial properties. There also silver nanoparticles have been used. Then even material for, say, defense forces, where special fabric is being made, which is very hardy because of the carbon nanoparticles or carbon nanotubes, which have been used to make them. So that even bulletproof things are being designed. So there are many areas where this nanoparticles have entered into life, even in medicine, tremendous use for say, developing anti-cancer drugs and all. Huge potential and huge usage they have. So now coming to the area nanoproteomics, what is it? So nanoproteomics, as Dr. Gupta mentioned, it's a marriage of these two areas. Proteomics is, as I mentioned, it's large scale identification of protein and also knowing their functions. Okay. The key point is whenever you talk of omics, you are working at large scale. And the necessity I've told you, the necessity is because a huge sea of information is available from our genome sequencing project. So we have to make sense of those. So that's why these areas, transcriptomics, proteomics, metabolomics, and many other omics area which are now coming up. You will see many terms which in literature which are existing. Basically, the main theme or main principle is working at large scale. Okay, so proteomics, when we are doing proteomics, there are some limitations, and I'll talk of those. And these nano, I have given an introduction of what is the meaning of nanotechnology and NP or nanoparticles as such. Now, how this shift has happened, or why there's a need of this shift again. So it's basically nanoproteomics, integration of nanotechnology with proteomics. And the reason why this was needed is because there are certain limitations of traditional proteomics methods. So this marriage of both has led to the next stage where those limitations can be kind of overcome. So nanoproteomics, this also I think Dr. Gupta mentioned that when you're having very small populations of cells. So now it's possible to do research at single cell level. Even single cell proteomics, single cell transcriptomics, single cell metabolomics, all these things are coming up now. So this has been made feasible because of this nanoproteomics. Okay, then these, and this is again, your nanostructures, their properties are basically, those are the things which have kind of facilitated development of this area. Now you have techniques because of presence of these nanostructures who have very excellent properties, say in case of, now there are techniques available which are called as nanoelectrophoresis, nanochromatography, because you have this nano material available, like for nanochromatography now, nano gold and nano silica and all these material is being used. And these columns, they have a property that they are very highly efficient. And you need just a small amount of matrix and you can do your job in very quickly. Time is also saved and reagents are also saved. So this is what I was telling, that this nanoproteomics, it allows real-time multiplexing. We need low sample volume as well as concentration, and that's why less reagent is needed. And the procedures are very sensitive and specific. And because you are working at low scale, your run times are also smaller, and you use miniaturized analytical system. Even your analysis material or analysis systems are very small. Okay, so, and this has been possible because of these properties which are mentioned on the right side of the slide, because of high surface area of these material, and their size and shape can be changed by changing conditions. So you can have, in fact, now a lot of sensors are being developed because you can change these nanoparticles. A slight change and you will have a different kind of property. And these things are being exploited to turn these nanoparticles into kind of sensors for various things, for sensing toxics, for sensing fertilizers, for sensing even your microbes, lethal microbes and general microbes. So, and your NP surface can be functionalized. Yeah, like you can attach different groups on them. And that is how they will then start, you can use them in some reagents. I think I have 1 or 2 examples. I'll describe that, what I mean. Enzyme immobilization is also possible on NPs, and that is how the efficiency of the system increases tremendously. When you have hooked the enzyme on NP, then you can reuse it many times. Similarly, electrical conductance is better in this material, so they have better efficiency. And even particle aggregation, this is also used because NP on aggregation, actually they change their property, because once they are in isolation, they will show them some property, but when they aggregate, there's a change in the property. Say sometimes they start emitting colored emission or colored light, or they change their color. And this kind of things are again being used to do, say, develop assay methods for enzyme. And by changing the property, like the product would be now colored. So if some binding happens, then it would aggregate and it will give you a different color. So the color intensity then can match with the activity. So certain assay, NP based assay, enzyme assays are also being developed. And all this is possible because of these extra properties these material acquire during synthesis or during formation. So this is how they are being used in proteomics for peptide fractionation, like gold nanoparticle. They are excellent, as I mentioned, they are excellent column packing material. So they are being used for making columns. Then your surface functionalization with different groups, that also helps in protein enrichment. You can enrich the small volumes of or small quantities of protein into huge quantities of protein, because the surface is functionalized in such a way that those particular class of protein or those proteins which are present in low amount, they'll sit on nanoparticle, and now this you can just scoop them out from that sample and you can elute them. So you can actually lay your hands on very low abundant proteins using these NP surface functionalized materials. Then in fact, sorry, you can also immobilize protein, as I mentioned, and that helps in increasing their efficiency. So trypsin has also been immobilized and it is being used for trypsinization. So the efficiency of trypsin increases several fold. Then for interaction analysis, the electrical and optical properties of these NPs are being used. Then the quantum dots, these are carbon-based nanomaterial, which are being used as labeling agent for detecting proteins in microarray, because these quantum dots, they show different colors, they emit different colors. So you can use them as tags for labeling your proteins in microarray, because when you are doing protein microarray, you have to detect them differentially. So if you can use tags, then it is the detection is made possible. So these quantum dots are being used as tags to tag proteins in protein microarrays. And then you have nanofluidics. This is basically working at nanoscale. Your whole instrumentation of proteomics now is working at nanoscale. So this area is called as nanofluidics. And basically you're using very small volume for here. So this is just one example of protein enrichment. Here your NP is hooked with zinc on its surface, and this zinc can specifically interact with phosphate groups. So that is how phosphoprotein enrichment is possible using this kind of material. And then you can identify these proteins by routine MS analysis. So you can basically enrich, so phosphoprotein identification or enrichment is possible. Then this is another example where a low abundant protein, which was present, it was enriched by using gold nanoparticle again. Some disease related protein in urine, it was present in very small quantities. So once you put this extract of this urine with this gold NPs, the protein from this mixture, they sit on the NP, they bind to the NP, and then you can extract these NPs and you can isolate this protein. And then when they compared these amount of these proteins, which were present in very low amounts, with protein precipitation procedure, there are certain procedures like acetone precipitation or TCA precipitation, by which you can enhance the amount of protein in your samples. So when they compared this with TCA precipitated samples, the results were better than TCA precipitated samples. So enrichment of low abundant protein is also possible using these nanomaterials. Then this is a miniaturized system for proteome analysis, the nanofluidics, which I mentioned. Here you are working at nanoscale. This is just a system where it's all automated. You don't have to, there's no manual interference. It's all run on wires. Nanoscale peristaltic pump, you can say, nano pumps. There's a group, and each step of the whole process of protein identification, right from loading your protein here in your auto sampler to then doing your digestion. This is your integrated trypsin digestion, which is happening in this chamber. So you put your sample here, and then you have nano valves through which this passes, and it is sent to this chamber where trypsinization happens. Then this trypsinization sample is sent to the next chamber for cleaning. And then from here, it is sent to the different columns where the purification or segregation of these peptides happen through nanopumps again. And finally, this is sent to the MS machine for identification. So different nanoscale pumps, they are working together and they are doing all the steps on their own. So it's fully automated and working again, as I mentioned, at nanoscale. So you need just 25 microliter of sample and you will get information out of that. So that is the maximum limit. You can even load less amount of sample. So these kind of setups are being used. And finally, this is for detection of coronavirus, where they have hooked the receptor for this spike protein on a carbon nanotube. And they have seen that on binding of the viral protein, as the receptor sees the spike protein or it binds to the spike protein, your nano sensor, this carbon tubes, their emission or the nano sensor fluorescence emission increases twofold. So this is being developed. This is a paper which was published in Nano Letters in last year. So they have shown that this could be a very good system to detect nanovirus, this coronavirus at nanoscale, because the amounts here, the detection limit is in nanometer, nanomolar. So very sensitive method, and they are trying to develop this as a detection method for coronavirus. And now shifting to the last part of the talk, protein corona. This is a term which is basically this tool, protein corona. It is a tool to understand nanoparticles. NP stands for nanoparticles, mechanism of action in biological system. And here I would like to just make few points. Over last say 1 or 2 decades, 1.5 or at the max 2 decades, many people like Dr. Gupta was also mentioning that they have also handled NPs, nanoparticles. They have done some work with that. So many groups and many research groups, they are engaged working with nanoparticles in some way or other. So what has happened, because a lot of people are working, so that means a lot of nanomaterial is kind of going into your atmosphere. Okay, but the mechanism of action of these NPs, whether it's animal system again or plant systems or microorganisms, how they are functioning, some information has started coming in last few years. But for plants, again, in last 2-3 years, I've seen some reviews and some papers about protein corona in plants. But actually, there's no information. Mechanism of action of nanoparticles in biological system, the information is very scanty, very less. So what we tried to initiate was this, that we wanted to understand the mechanism of action of these nanoparticle and the system which we are using is Brassica junctia, mustard. So we are using mustard at the system, and we are using gold nanoparticle, and then we are trying to understand this protein corona. Now, what is this protein corona? I'll tell you. This is a tool which we are using. So I'll straight away go to the next slide. So this is what protein corona is. This is your nascent NP. As soon as nanoparticles, they enter into the biological system, various macromolecules, not only proteins, your lipids or carbohydrates or other small molecules, they start binding onto these nanoparticles. Okay, and in span of few hours, they are no more naked. They are all covered. They are all bound by different macromolecules. So especially protein corona is focus on analyzing protein which are binding onto a nanoparticle. Okay, so we are trying to understand which protein are binding to nanoparticle, and this again there would be a variation on depending on which nanoparticle is being used. There are variable factors which control this NP corona, this protein corona formation. So why we are doing this? Because we once we know what kind of protein are interacting with the nanoparticle, we can probably guess what functions they have. So what would be the effect on function, and then we can measure that function. So that was the whole idea with which we started this work a few years back, just 3-4 years back. And as I mentioned this, so what happens when these proteins are interacting or sitting on or binding to this NP? Initially, within very few hours, you have some proteins which will bind to this NP, and this initial binding is called as, this constitutes soft corona, the initial binding which happens. And then later with time, as the incubation time increases, these proteins are replaced by other proteins, and you have formation of hard corona, which is the final form of your protein corona. So now you can take out protein from this NP, and you can identify them as normal identification is done. Only difference is once your NP is bound by your proteins, now you separate or trypsinize this whole thing. You take the whole thing and you trypsinize it so that your peptides comes out into solution. And then these peptides are subjected to MS identification. So this is what we have done. Idea or the basic thing was to understand the biological mechanism. Once the NP enters, what happens to it in the biological system. And as I mentioned, there are several factors which would affect this corona formation. And these factors are your NP based also, and your system based also, like what system you're trying to analyze. So when you talk of nanoparticle based features or properties which will affect corona formation, your NP size, its shape, its surface chemistry, the pH of the reaction, the protein amount which you're using for binding, the incubation time, temperature, and what type, because each type of NP would bind to different type of protein. It won't be same. Okay. So these are all variables which you have to take care of. And when you talk of system, again, what system had what kind of protein? So that would, again, the binding would depend on that also. So these analysis, protein corona analysis, what it is used for is, it is used for NP-assisted drug or nutrient delivery. It's used for suitable ligand discovery, like which proteins are binding. So then you can even use those bound protein for something else. So ligand discovery is possible. Then mitigation of toxic effects of pollutants. And here, basically, what happens, your NP is there. So pollutants, certain pollutants they would have, or even you can even change the surface. You can bind certain groups on the NP, and so they then definitely specifically bind to a class of toxins. So, and then you can just remove this NP, so your system is clean. So various effects of different pollutants, whether it is in soil or it is in water, those studies are being done where these NPs with these functional groups are being used. Even for biomarker discovery, they are being used. And the last point, improving agriculture yield. This is very far right away. We are just, as I mentioned, we just initiated some work and it takes time. So we are still to give very concrete, but we have done some initial experiment. I'll share those with you now. So these are the challenges which we faced when we started this work. Protein corona, PC, is extremely sensitive, as I mentioned the list, to several NP specific and environment specific factors. Therefore, reproducible data generation is difficult. You have to repeat your experiments under very controlled conditions several times to get repetitive results. So this problem we had faced. Then again, your NP has to be of the same size for which you are interacting, which you are studying interactions. If you're, as I mentioned, if your size changes, interaction also changes. So your NP has to be of uniform size and of uniform properties. So that is the very important or tight control step. Then you have discovery, you have difficulty in recovery of analyzable NPs or good amounts of NP out of the system. And this is again a big hurdle which we still have to overcome. When we are trying to put this NP in plant systems, recovery is very poor. We are not able to yet overcome this hurdle. So, and that is why, because of this hurdle, the third point, there's no information available for in vitro, in vivo comparisons of the proteins which would bind, and you can do a comparison and you can relate to in vivo situation. So this is again a lacuna which is still not, we are still struggling with it, because in vivo we are not able to get enough amounts. In vitro we have got some information, so we have tried to derive something out of from them, that information. And this last point, as I mentioned, this is still to be, it's very far. Right now I can tell you that a link between information, PC information, protein corona information, and their translation for better plant health or better plant yield, it is a far-fetched dream till now. And this last point is also very important. Right now, very few people are working on this area, so you don't find people who can handle this work, or so workforce is scarce. So when we started this work, we just asked us few simple questions. And these are very obvious questions, and the answers was to some extent were obvious. But when you do research, you have to prove things experimentally. So our first question was whether when we do comparative corona analysis, whether the results would be different and it will behave differently. These were some very basic questions, because there was no information. Definitely from human system, there is some information and some clues are there. But for plant systems, I see some paper in last year. Some paper now are appearing on protein corona. But when we started this work, no information absolutely. So this was the initial question which we asked ourselves. So for this, your gold nanoparticles were synthesized. And as I mentioned, the procedure is to be such that your gold nanoparticles, they are of almost similar size and of similar properties. So your conditions have to be tightly controlled, and it's possible to like characterize these properties of this nanoparticle. Their size you can characterize, the type you can characterize. There are various procedures which are available, standard procedures which you can follow, and you can characterize your size, shape, and charge, and your even the type of NP which you have synthesized. So for that, your routine UV spectrometry, then TAM, your electron micrographic transmission electron microscopy microscopy is done. Then XRD spectras are taken just to identify the element for which you have formed your NP for, because each element would have its signature XRD spectra. So these are all characterized properties which can be done using different techniques. And I think I'll skip this. This is just to the experimental procedure, just to isolate these NPs from extracts. So basically it's centrifugation under controlled conditions. Again, your speed and time, all this has to be optimized once you incubate your extract with your NPs, and then you just look for the pellet of your NP with protein sitting on it. And when we characterize the protein corona, what we could see that the size of the NP, this is your initial size, about 25 nanometer, 21.5 plus minus 3.3, and it increased to 55.75 on binding of protein. It almost became double, slightly more than double. And this is just a picture to show, sorry, this is just a picture to show the scanning picture, just to show the NP with protein sitting on it. And this is the other ways by which these NPs can be characterized. As I mentioned, UV spectroscopy is one usual way. And here, this is the actual experiment where we compared the binding of proteins from crude extract with nuclear extract. The part A is your crude extract and part B is your nuclear extract corona. Okay. So you can see distinctly that in the part A, you see two different types of lines in your UV graphs or spectrums. First, you see a peak, R1, and second, these are slightly broader lines with no peaks, R2. So in case of crude, you could see two distinct phases, and these two distinct phases, they show soft corona and hard corona. Okay, the R1 is your soft corona and R2 is your hard corona. And if you see compare the result with in the picture B, then this is your nuclear corona. So here you don't see that kind of phasing. Two different phases you don't see very clearly. You just see a kind of increase in intensity. The peak is enhanced. If you see here, this one, this is soft corona and this is your hard corona. Okay, so the intensity increases there. The type of peak is same. So there's no difference in the quality. Quantity is only changing. And this was again established by doing SDS page of these protein coronas. And here you can see that the protein profiling in case of crude, you can see two different types of profile from 2 to 8 hours on the top, 2H, 4H, 8H is your time. 2 to 8 hours, that is your soft corona. And after 8 hours, 16 hours to 36 hours is your hard corona. So you can clearly see this what peaking which we are seeing differentially, the same thing is seen on the SDS page gel also. And in this case, only the intensity difference. Same proteins are binding, but the intensity is less, and that is how you see in the peak also only the difference in intensity. So that means in case of crude extract, you were getting formation of soft corona as well as hard corona. So that means distinct proteins are binding in soft corona and hard corona. While in nuclear extract, this binding was kind of more uniform. And this is what we could see from these two experiments. And we went further so we tried to identify these proteins. As I mentioned, we sent them for MS identification. And when we looked for the protein which are binding to these protein coronas, if this is a comparison of the first part is in crude, the soft and hard corona, light bars are soft corona and the dark bars are hard corona. So you can see that this is your soft corona, these light bars. Okay. So the soft corona, the binding of the proteins, the molecular weight wise, you see they are having a broader range. Proteins from 16 to 30 kDa, then 31 to 45 and 46 to 60. That means 16 kDa to 60 kDa. They are binding to this protein corona in case of crude extract. While if you see the parallel, this one, which is a nuclear extract, only a narrow range of size of proteins are sitting or binding to this particular corona, both in soft and hard. And they belong to 10 to 20 kDa category of size of proteins. So in short, you can say that wider size protein, they bind to your nanoparticle from crude extract, while in nuclear, very narrow range, 10 to 20 kDa protein only were binding. Okay, so size wise, there was a distinction. Relatively broader range of protein were binding to your NP in case of crude, very narrow range for nuclear extract. And we also looked for some differences in charge wise. Say, can we see some differentiation with regard to charge of this protein when they are binding or to the these coronas? So again, a comparison of crude and nuclear extracts was made, and it was what you can see here on the top bar diagrams. This is soft, the light one, light bars, and the dark ones are the hard ones. So when the soft corona is getting formed, you have a widespread charge, and in fact, more protein with positive charge or on alkaline side, they are binding to soft corona. But as hard corona is formed, you have more of negative protein. They are peaking more, so they are more in number. But again, on nuclear, soft and hard corona, there was no such distinction. Similar kind of charge distribution was seen for all the protein. So this was another information which we could derive from this experiment, that charge plays a role in case of corona formation in crude, but in nuclear, it's almost the same. In hard and soft corona, the proteins are almost of the similar charges. There are no great distinctions. And then we went ahead to know the cellular location of these proteins. We compared that, which cellular part these protein belong to. When you compare the soft corona versus hard corona from nuclear and crude extracts, so that you know that which type of protein or type of protein from which organelle or which part of the cell are sitting more on, say, nuclear corona and crude corona. So when we compare the soft versus hard, so soft is the early one and hard is the late one, the final one, you can say, the final protein which are sitting on the NP. So what would we see that in this case, when we compare location wise, if you see the soft corona here, this is nuclear, light, again, here the labeling is different. Nuclear is your light one and crude is your dark one. So when you see the nuclear proteins, definitely it's a nuclear extract. So your nuclear proteins would be more in the soft corona. Okay, so you see almost 51% protein, the nuclear protein, and relatively very less amount of crude protein. Definitely it's a nuclear prep, it's purified to a certain extent, so you will see that distinction. Okay, and ribosomal protein were almost in same, they were sitting tight on soft corona in both nuclear as well as crude, the second high bar, 34 and 42. Then you have certain other categories which were there, but the main change which happened in hard corona, like there was a shift now. Soft corona, some proteins are sitting, now they are replaced by other proteins which have probably higher affinity. for the NP. So they will replace the protein which are sitting in soft corona on the NP. So when we looked at these bars or these values, then some changes which we observed were chloroplastic protein were high in case of hard crude corona. Okay, so that means initially they were less, they were 15%, now they have become 28.5%. So they have better affinity even for hard corona, so they can bind better to the NP. Similarly, your even cytosolic protein, some enhancement was there. But if you see in hard corona, the nuclear ones, they have decreased to some extent. They have from 51, they became 38. Even ribosomal, they fell by some amount in case of hard corona of nucleus. But in case of crude, there was a drastic change in binding of ribosomal protein to NP with crude extract. And there were some differences which we could observe, like cell wall protein, they appeared in crude corona, hard corona. Then peroxisomal also, there was some enhancement in binding of those proteins. Similarly, membrane proteins slightly better in case of crude hard corona in comparison to nuclear corona. And the reason again could be because of the extract being used. Because crude extract would have proteins from all different sites of the cell. But nuclear would have relatively more number of nuclear proteins. So probably that's how you can see differentiation or segregation category wise in this case. Now the next point was, these are all routine studies which are generally done this way only. Next was to know functional classes to which these proteins belong to. So we could see almost, these are the different functional categories on this panel you see. So the dotted bars are important because these are the proteins which were kind of coming more in your crude hard corona. Leaf senescence, cell wall organization, then TCA cycle, glycine biosynthesis, glycolysis, although they were almost similar, but they were also coming. Photosynthesis. So if we join everything together, almost 25% of the protein in functional categories, they belong to energy yielding pathways. So this is a significant piece of information which we are carrying forward now. We are trying to analyze the photosynthetic efficiencies of these plants after NP treatment. Another question which we asked ourselves was whether these enzymes which are binding to these nanoparticles, whether they are active, whether they retain their activity. So we just randomly did the most abundant one which was sitting on these NPs. We did their activity measurement in crude extract as well as in hard and soft corona. And what we could see here, these are the two proteins. The light bar is for malate dehydrogenase on these bars, and the dark one is fructose bisphosphate aldolase. These are the top targets which are coming on these NPs. So in case of soft corona, this is basically total leaf soft corona, because now we went to analyze the leaf proteins. Initially, after doing the different cellular components, we went for total leaf protein analysis, and they're binding with the protein corona, again with gold nanoparticle. So what we are seeing here, that this particular fructose bisphosphate is missing in soft corona, but it appears in hard corona. That means it is better binding in hard corona, or it binds better later. Initially, it is not bound, it is not having activity, or probably its amount is also increased. So the important point is that the proteins are active. They are showing activity even in the bound form on these NPs. Another experiment which we did was Rubisco was also coming as a target. So we did CD analysis to know the change in conformation of Rubisco. And it was again an encouraging result, because in the Rubisco, which was bound to the NP, this red line, it showed higher beta sheet dominated structure. And higher beta sheet dominated structure means more stabilized protein. It provides stability to protein. So that means Rubisco, when it was sitting on NP, it was more stable comparatively to the control. And this is just purified protein. This is a FTIR spectrum of, CD spectrum of your purified Rubisco. And this is your Rubisco sitting on your NP, bound to your NP. So that was also encouraging. That means conformation is also being stabilized, because at that moment, actually, normally for Rubisco activity, you need radioactive materials. So we were not able to do radioactive reactions. So we did this. We just wanted to analyze what is the effect on the conformation of protein. So this was also a positive result which we obtained. And this is what we can conclude right now from these studies. Temporal evolution of PC, that means conversion of PC from soft to hard corona, is influenced by PI of proteins. Initially, the basic protein or the protein with positive charge, they bind in more number, and eventually they are replaced with the protein with negative charge or acidic proteins. Then we also observed, and this I have already mentioned, about 25% of the protein, they belong to energy related pathways. So the question is whether we can use these proteins to manipulate energy pathways in brassica junction. And then somehow, so for this, we are doing some experiments where we have supplied these NP to brassica plants in pots and field, and we are measuring the photosynthetic capabilities of these NP treated plants. And then we would try to relate it with the results we have obtained. And we are getting some positive results on that line, but still to be confirmed. Few things still needs to be confirmed. So I'm not showing with full, saying anything with full conviction. And besides this, there was another observation that almost 10% of the corona proteins, they were proteases, which were binding to the NPs. So, and normally proteases, they have been shown to be involved in senescence or cell death. So whether they have any role to play or any role in preventing or controlling leaf senescence or cell death, this is another question which we are trying to address. And this I've also mentioned that I've already mentioned that the secondary structure of Rubisco was stabilized or better because it was showing more beta sheets. And this work was published in Plant Physiology and Biochemistry. And I think this is the last slide. Just few concerns or few questions or few, what should I say, the future experiments which we want to do in this area is we want to do sequential interaction protocol. What is that? Right now, what we have done, what I have shared with you, this is all in vitro. We have made extracts, we have incubated NP with those extracts, and we have tried to analyze which protein are binding. So first, very important question is whether in vivo also same thing would happen. So we have been trying to do those in vivo experiments, but as I mentioned earlier, we are not successful till date. So we are still working on that system, and there's no report in literature about in vivo protein corona formation in plants and analysis of protein corona in vivo in plants. So it's a challenging area of work in that sense. So sequential, what I was trying to say is that right now, as I mentioned, we have just used fruit extract and nuclear extract, and we have tried to analyze the protein which are binding. But in a cell or in biological system, when NP enters, it will enter through your cell wall, then it will go to apoplast, cell membrane, then it will go to cytosol and different organelles. So what happens to NP once it enters into biological system? Because it won't be same. What we have done is it's a test tube experiment. It's an in vitro experiment. But in vivo, because it will interact with the proteins probably in cell wall, then in apoplast, then cell membrane, and it will be a different kind of protein corona. So what happens, how that protein corona develops once it enters into the plant system? That is a big question. And for that, we are planning some experiment where we are trying to do this in sequential manner. Like we will analyze the protein corona at one location, then we will try the protein corona at different second location, then the third location. So you will have to do it in a sequence. And that will give us definitely a different kind of information. So that those experiments are being planned right now. And then the last point I have mentioned to you, that we are analyzing photosynthetic yield of these plants after NP treatment, so that we can correlate the information from the two sides. And that is where right now we have done some enzyme activities and all. We are getting some encouraging result, but it is yet to be kind of shared. And these are the students. This is botany department, a picture from the botany department. It's called Araucaria lawn, a famous place in botany department. And these are some people. This is Satya Chaurasia. He has done all the work on protein corona. She's Meenakshi. She's a student of your college. Deshbandhu, she has just finished her PhD with me. She worked on cuticle proteome, analyzing cuticle proteome. Again, the system is Brassica junctia. She is Bhavna, the third student in pink blazer or sweater. She is worked basically on Seebuckthorne antifreeze protein, done wonderful work, has I think 10, 11 publication from her thesis. And then next is Ankita. She has worked on nitrosilation in, again, system is Brassica, nitrosilation in apoplast. Then the next person in blue sari is Jasmeet. She is the one who is till date my best student. She is the one who established nitrosilation work in the lab. Then Shruti, she is working on diascoria, and she has also done good work. Again, all of them, they are using proteomics tool to understand biology. Then Priyanka, she's still continuing. Yai, she is also continuing, and the last person has left my lab. She's in some other university now. And besides this, we also conduct workshops on proteomics. This was the first one, which was done in 2013, and in this coverage of this was done by, I think it is a proteomics journal. The paper was published there, and this is one of the organization, International Plant Proteome Organization. I am a country representative for that, so they covered this in Plant Proteomics Journal. And this is some glimpses of these workshops. And you can see, I don't know if you can see Shantanu here. He also attended somewhere here. His face is not very clear. You can see him here. And there are other people from colleges of Delhi University, as well as people from outside Delhi. All over India, they come and they attend these workshops. And we do this workshop very seriously. But unfortunately, since last 2 years, we have not even been able to do. We do this alternate year. Last one we did online, not a workshop, workshop come conference, because physical was not possible. So thank you for your kind attention. And funding basically I've got from DBT and Delhi University R&D grant and CSIR for most of the work which we do in our DRDO, and several funding agencies, they have supported our work, but this particular one was particularly by DBT and a bit by IOE grant. So I think with that, I'll stop sharing my slides, and I am ready to discuss and have any questions being, or if any discussions or queries you have. Sorry, I'll just have a bit of water and then probably we can continue. Yeah. Am I audible or not?
Yes, ma'am.
Okay, I thought I lost connection because these issues nowadays it's become so bland and dry presenting online, because yeah. So you are Chaitanya.
Yes, ma'am.
You are also a
I'm vice president.
society member.
Yes, ma'am.
Okay. I think good to have these societies, because they do a lot of work for students. A lot of awareness and learning happens by this, and you are also trained in kind of handling things. So I think it's good for students, these efforts.
Yes, ma'am.
Yeah, Chaitanya, please.
Ma'am, there was some network issue from Neha Lohan, ma'am. Ma'am, can we?
Are there some queries from students or some questions or?
There are questions, ma'am. There are questions.
Okay.
Neha had to do this part, ma'am. Chaitanya, you can take over, Bache. If she has
some
lost connection, you can take over.
Okay.
Yes, ma'am.
Yeah.
It was a great pleasure to listen to you, to your wise words, and learn so much from it. Thank you so much, ma'am, for such an inspirational and interesting talk. We are very fortunate to be a part of this webinar. Your years of research, your depth of understanding, and your ability to present the subject in such an interesting way help us to understand in a much better way. Our enthusiastic participants have asked some interesting questions in the chat box. With your permission, ma'am, I would like to present them to you.
Sure. Please go ahead.
Thank you, ma'am. Ma'am, the first question is, what is the difference between nanoparticles and quantum dots?
Quantum dots are also a type of nanoparticle. They are basically carbon-based nanoparticles. Okay. But their properties are slightly different in the sense quantum dots, depending on their sizes, they emit different kind of fluorescent light. So they mostly are being used as probes for probing for like they can be linked to certain, say in case of, I mentioned one of the application on protein microarrays. Like you can hook these and you can use them as tags to identify different proteins. So basically they're also a type of nanoparticle, but slightly different properties, and they are carbon-based nanoparticles.
Thank you, ma'am. And the next question is, what are the applications of modern nanotechnology and what are its percentage in different fields? Percentage of actual achievement in different fields.
As I mentioned, I just mentioned few things, few applications which are commonplace or which are entered our life. There are many applications. I touched on them, say about fabric coating, then in agriculture, nano pesticides, nano fertilizers, then in like I mentioned about purification of water, pollutants are being removed. Okay, similarly, toxics are being removed. And if you talk of biomedicine,
yes, ma'am,
they are being used extensively for diagnostic, for treatment, for anti-cancer treatments, for even dresses, your dressings of containing antibacterial properties are being made now with nanoparticles. Then recently, if you read newspapers and all, they have made a nasal spray for corona. That is also NP based. Even a few years back, there was another application where a filter was made by IIT person, where it was basically very small pores that also contain NPs. It will all the pollutants, they'll stick to the filter. You can put that filter in your nose. It's what is a nasal filter, and you can be prevented by the bad pollutants which are present in the air, because it will adhere all the pollutants to on that filter, and then you can change the filter. So those kind of applications, many applications, like probably thousands of them. So telling percentage wise, it will be very difficult, because this field has now taken over everywhere, whether it's agriculture, biomedicine, food, everywhere. You see many applications. If you just scan the literature, many applications are there.
Yes, ma'am. Thank you so much, ma'am, for this wonderful answer, ma'am. And the next question is, what are the advantages and disadvantages of proteome analysis?
Okay. Advantages, you can study proteins in big numbers. Like you can know the proteome of a particular cell or organelle or tissue or an organism, if you're talking of unicellular organism, in a few attempts. That is a plus. But the negatives are, you need a lot of infra. You need MS machine, which is in crores. The cost of a MS machine is in crores, but definitely you can outsource your work. And you need a bit of expertise to do. You have to learn those procedures, and then only you can do them. But information, you get a huge amount of information, and then you can make sense of that information, in the sense you can do comparative analysis, you can know the functions of protein, as I mentioned in my presentation, that normally when you are doing annotation of genomes, we have a lot of information which we don't know, a lot of coding sequences which we don't know what are they coding for, what are the functions of those proteins. So that kind of information, by doing proteome analysis, we can dig out that information, and we can help annotation of genomes by providing that functional input from proteome analysis.
Thank you, ma'am. Ma'am, the next question is, how is bioinformatics used in proteomics?
Okay. I explained to you about one method called PMF.
Yes, ma'am.
है ना? PMF is one of the method by which you can identify proteins. In that, there are two parallel things which you do. On one side, you take protein, digest them, give it to MS machine, you get MS spectrum. On the other side, what you are doing, you have sequences in databases. You are using softwares to first you get ORF, the reading frames from there, the protein coding sequences. Then using softwares, you get the peptides. Protein cutter is a software which is used, which tells you how many peptides can be produced by that particular genome, coding parts of that genome. And all those peptides are listed in the database. So here you have got the spectrum. There you know the sizes. यहाँ पर आपको पेप्टाइड मिले पेप्टाइड कटर से, उनका आपको मासेस भी मालूम है, मतलब पेप्टाइड की लिस्ट है और मासेस भी हैं. Now to identify protein, you basically match these two, the experimental information which is coming and the database information which is coming, and that is how you identify protein. So this is very vital. If you don't have this information has no meaning. You have to have this information to identify the protein. So that is the significance of databases and software in proteomics analysis.
Thank you so much, ma'am. Ma'am, the way you explained all these questions is so remarkable, ma'am.
I hope students
Yes,
are
ma'am.
able to imbibe something. That is the whole purpose. कि थोड़ा सा
Yes, ma'am.
excite हुए, थोड़ा सा समझें.
Yes, ma'am.
I
Ma'am,
just
the
hope
next-
it's not gone overboard. कुछ समझ में आया है कि बिल्कुल ऊपर से चला गया है? बोर लग रहा था? Very
No,
frank
ma'am.
question. समझ में आ रहा था कुछ?
Yes, ma'am.
अच्छा, चलो हाँ. What is the next question?
Ma'am, the next question is, ma'am, are there any ethical issues of nanotechnology? And if yes, how they can be solved?
Yes, actually, ethical भी है और biosafety issues भी है. And this happens with all technologies. जब हमारा microwave आया था, we were so excited. Oh, microwave आ गया, now we can heat food and it's so convenient and all. But अभी पता लग रहा है कि even microwave heated food has problems. It causes cancer.
Yes,
So
ma'am.
usage के बाद ही पता लगता है कि क्या है. Similar stand is for NPs now. We don't know the biosafety aspects of that because उसके लिए ना you have to formulate guidelines on usage. Many people are using them, but we still don't have the guidelines. Recently, I think last year, DBT and DST, they have formulated some guidelines, but they are strictly to be implemented. There are rules to be made that you have to do this way. Otherwise, biosafety issues are a real concern, and even bioethics can also be a concern in NP work. If you are not going in an informed manner, then it can be actually dangerous. So I think that is a big, what should I say, a big bottleneck in NP research. And that is why, although a lot of things are being done in labs, but it is not getting translated because rules नहीं है अभी, legislation नहीं है. नहीं तो जो इतनी सारी चीजें बन रहे हैं, जैसे अभी recent example is of nano urea, which was put to use. It is now being used. Nano urea is being used, and it is used in very less amount, and it is very cost wise also it's very good for farmers. But ठीक है, उसको रिलीज कर गया है, हमने श्रीलंका को भी दे दिया है, लेकिन अभी भी जो रूल्स है ना कि नैनो मटेरियल को यूज करने के लिए, जैसे हमारे हर चीज के रूल्स होते हैं, इवन रूटीन बिजनेस हम करते हैं वहां भी रूल्स होते हैं, दीज आर द रूल्स. सो यहाँ पर भी अगर हमें एनो बेस्ड चीजों को अगर मार्केट में लाना है, सो वी शुड हैव वेरी क्लियर रूल्स ऑन दैट. तो वहां पर बायोसेफ्टी इज वन बिग कंसर्न, क्योंकि बायोसेफ्टी ही अभी बहुत अच्छी तरह से समझ में नहीं आता है, लोगों को मालूम नहीं है कि कैसे, हाउ टू यूज, हाउ टू कंटेन एंड हाउ टू prevent, because now what is happening, so many people are working with nanoparticles, so a lot of accumulation of these nanoparticles is happening in soil, in water, in air, because they are so small in size, वो disperse air में भी हो जाते हैं, because very small, heavy नहीं है, तो वो air को भी pollute कर रहे हैं. So this is a big concern, and this is something which is actually kind of restricting the usage of nano, although there are many usage, as I mentioned, they are being used. But this is one issue which needs to be tackled or to be handled.
Thank you, ma'am. And the next question is, ma'am, which one is more complex, proteomics or genomics?
Definitely proteomics. Like in what context you was asking, because genes जो होते हैं ना, जैसे मैंने आपको बताया कि Arabidopsis में there are about 25,000 genes.
Yes, ma'am.
Human 30 something gene, 30,000 genes. ह्यूमन वी कंसिडर बहुत ही एडवांस है evolution wise, no? Plant Arabidopsis is such a small plant which can be grown in labs, and इतना सा होता है, उसमें flower भी आता है. And if you see human, definitely complex है. लेकिन अगर आप प्रोटीन्स देखें ना, आगे जीनोम पे उतना वेरिएशन नहीं है. नंबर ऑफ जीन्स, दे आर ऑलमोस्ट सिमिलर और वेरी वेरी नियर टू ईच अदर. लेकिन प्रोटियोम पे जब आते हैं ना, या प्रोटीन्स पे आते हैं, बिकॉज डाइवर्सिटी कम्स बिकॉज ऑफ पीटीएम्स, बिकॉज ऑफ स्पाइस वेरिएंट्स, बिकॉज ऑफ पोस्ट ट्रांसक्रिप्शनल चेंजेस. सो एंड यू हैव मल्टीपल आइसोफॉर्म्स ऑफ प्रोटीन. सो जो प्रोटीन नंबर है ना, वो फ्रॉम फ्यू थाउजेंड इट बिकम्स टू प्रॉब्ली लैक्स. सो प्रोटीन इज डेफिनेटली, प्रोटियोम इज मोर कॉम्प्लेक्स देन योर जीनोम्स.
Thank you, ma'am. So the last question is, ma'am, what are the major components of the proteogenomic study?
Major component. You basically, sorry, in proteogenomic studies, हमारे पास नॉर्मली इंफॉर्मेशन जो जीनोम का है वो सारा कहाँ है, डेटाबेसेस में है ना?
Yes, ma'am.
तो सीक्वेंसेस वगैरह सब वहीं पर है. तो एक तो आपको डेटाबेस चाहिए, फिर आपको सॉफ्टवेयर चाहिए जो कि आपका स्कैन करते हैं पूरे सीक्वेंसेस को, और उधर से आपको उसको, जैसे मैंने बताया, ट्रांसलेट करके, एंड इंफॉर्मेशन अबाउट पेप्टाइड्स आल्सो कैन बी डिराइव्ड. सो ये सब आपको डेटाबेसेस चाहिए, आपको सॉफ्टवेयर चाहिए, आपको जीनोम इंफॉर्मेशन चाहिए, और आपको प्रोटियोमिक्स का भी सेटअप चाहिए जहां से कि आप प्रोटीन्स आइडेंटिफाई कर पाए, एंड देन यू कैन रिलेट थिंग्स. सो दोनों तरह का इंफॉर्मेशन चाहिए आपको. दैट इज द रिक्वायरमेंट फॉर प्रोटियोजेनिक्स. या,
Thank you, ma'am. Ma'am, there was one more question, ma'am.
बताइए.
Ma'am, what are the future perspective of proteomics in medical field or in other field, ma'am? And how it is useful, ma'am?
In medicine also, proteomics is already being used for detecting biomarkers for diseases, okay?
Yes, ma'am.
For developing anti-cancer agents, for developing, for even analyzing the disease state and the normal state, and then looking for the pathways, and to understand the mechanism, how the disease is happening, how the disease progression is happening, even in diagnostic, because we can use markers even for diagnosis. So several markers at diagnostic, then for progression of the disease, and even for treatment. So all stages of medical profession, they are being used. And again, a lot of papers, thousands of papers. Plant proteomics is relatively later, but if you see human proteomics or disease proteomics, बहुत information है. Lot of information is available, and it is being used in a big way.
Okay, ma'am. Thank you, ma'am. It seems as if all the questions have been taken up. Thank you, ma'am, for answering all of the questions patiently and meticulously. Before moving forward, I would like to invite Muskan to present our guest speaker, Professor Renu Deswal, with a token of gratitude and appreciation in the form of the certificate and memento, to sharing her valuable knowledge on the topic proteomics to nanoproteomics.
Thank you very much. Thank you.
Thank you so much, ma'am. Now, I would like to invite Jennifer David to take over and carry this event forward.
Thank you, Chaitanya. Coming together is a beginning. Staying together is progress, and working together is success. Having said that, I would now like to introduce everyone present here to our newly elected office bearers and council members for this session. Starting with Ms. Priyanka Barua from BSc Honours Botany 3rd year has been elected as the President of the Society. Mr. Chaitanya Dhussa from BSc Honours Botany 2nd year has been elected as the Vice President. Ms. Tanya Saxena from BSc Honours Botany 2nd year will be the Treasurer for this session. And Ms. Neha Luhan from BSc Honours Botany 3rd year has been appointed as the Secretary.
Congratulations, everyone. And you are doing well.
Thank you so much, ma'am.
Mother Teresa rightly says, I can do things you cannot, and you can do things I cannot. And together we can do great things. As our Botanical Society will be incomplete if we mention only the office bearers without speaking of the class representatives and volunteers upon whom they can always count on. I would like to introduce you all to our class representatives and volunteers. Class representatives of Botany Honours 3rd year, Mr. Aditya Prakash and Ms. Tanya Bangalia. Class representatives of Botany Honours 2nd year, Ms. Naisa Hanglu and Mr. Sandesh Kumar. Class representatives of Botany Honours 1st year, Ms. Devanshi Tiwari and Mr. Amarnath. Council members from Botany Honours 3rd year, Artika Kumari, Dipali, Jennifer David, Muskan Makhija, Riya Paul, Shifali Sharma, Suhani Kumar, and Yukta Verma. Council members from Botany Honours 2nd year, Harshveer Chauhan, Krishna Yadav, Kirti Anand, Madhu Smita, Sakshi Navani, Surbhi Garg, and Vishakha Sagar. Council members from Botany Honours 1st year, Aditya Raj, Bharti Yadav, Dipanshu Chandel, Mansi Sharda, Amiksha Sewal, Riya Vashne, Sambhavi, and Shrestha Tyagi. Last but not the least, it gives me immense pleasure to introduce you all to our Society Advisor for this session, Dr. Sunita Malik. Due to the current situation of the pandemic, I humbly request the parents of council members Due to the current situation of the pandemic, I humbly request the parents of council members and office bearers to bless them while they wear their badges. Soon after this, we will have the oath-taking ceremony, which will be led by our newly elected president, Ms. Priyanka Barua from BSc Honours Botany, 3rd year.
I, Priyanka Barua, President of Botanical Society.
कुछ पाने की हो आस आस कोई अरमा हो जो खास खास आशाएं हर कोशिश में हो बार बार करे
I'm Chaitanya Dussa, Vice President of the Botanical Society.
तूफानों को चीर के मंजिलों को
I, Neha Luhar, Secretary of Botanical Society. I, Tanya Saxena, Treasurer.
I, Tanya Bangalia, class representative of Botany Honours 3rd.
I, Aditya, class representative of 3rd year Botany Honours. Thank you.
My name is Sandesh Kumar and I am a class representative.
I, Naisa Chandu, class representative second year.
जन्नत से मुलाकात हो, पूरी हो तेरी हर दुआ।
I, Amarnath, class representative, first year.
उम्मीदें हंसे की, अब मुश्किल नहीं कुछ भी, नहीं कुछ भी।
Divyanshi Tiwari, class representative of Botany first year.
I, Dipali, council member of Botanical Society.
गुजरे ऐसी हर रात रात हो
I, Shifali Sharma, council member of Botanical Society.
लेकर सूरज से आग आग
I,
गाए जा
Muskan
अपना
Mukhija,
राग
member of Botanical
रहा
Society.
कुछ ऐसा करके दिखा, खुद खुश हो जाए खुदा। आशाएं
I, Suhani Kumar, council member of Botanical Society.
अब मुश्किल नहीं
I, Jessica Davis, I'm a member of the council of the Botanical Society.
I, Priya Paul, council member of Botanical Society.
I, Artika Kumari, member of Student Council.
I, Vishaka Sagar, council member of Botanical Society, Hindu College.
I, Sakshi Nawani, member of the Botanical Society.
कुछ पाने की हो
I,
आस
Kirti
आस,
Anand,
कोई अरमान
volunteer
हो जो
of
खास
Botanical
खास
Society.
आशाएं। हर कोशिश में हो बार बार करे दरियाओं को आर पार आशाएं। तूफानों को चिर के मंजिलों को
Here I am Shambhavi from first year, member of the Student Council.
उम्मीदें हंसे की, अब मुश्किल नहीं कुछ भी, नहीं कुछ भी। आशाएं
I, Riya Vaishnav, council member of Botanical Society.
अब मुश्किल नहीं कुछ भी, नहीं कुछ भी।
And I request all the council members to switch on the camera and mic. We, the member of council,
The member of council
The member of council
solemnly swear
solemnly swear
to act for the benefit
to act for the benefit
of this council
of this council
and to remain goal-oriented,
and to remain goal-oriented,
motivated and dedicated
motivated and dedicated
to the working of this council.
to the working of the council.
We swear to help
We swear to help
and promote the development
and promote the development
of the council
of the council
and support it to its
and support to its
best of our abilities.
best of our ability.
best of
We promise to be
We promise
to do always enthusiastic
Always enthusiastic
and have an optimistic attitude
and have an optimistic attitude
towards our responsibilities
towards our responsibilities
as a council member.
as a council member.
as the council.
We will abide by the rules
We will abide by the rules
and regulations of the society
and regulations of the society,
and will uphold its glory
and will uphold
its glory
and dignity.
and dignity.
And
Thank you, everyone.
A mentor is someone who sees more talent and ability within you than you see in yourself and helps bring it out of you. Now, I would like to invite the Botanical Society advisor for the session 2020-21, Dr. Sunita Malik, to please say a few words.
Thank you, Tanya. On this bright, sunny Friday afternoon, a very warm welcome to Professor Renu Deswal, who is the galaxy of knowledge. Vice Principal, Professor Kamal Kumar Gupta, who is motivating and ever inspiring. Esteemed alumni present here, who are the pillar of Botany Department. My fellow colleagues and bundle of talent and budding scientists are dear students. It gives me great pleasure to congratulate and welcome the newly elected Student Council of Botanical Society of Deshbandhu College for session 2021-22. I am sure the Student Council office bearers will continue to demonstrate leadership and sophistication for maintaining a striking balance between academics and execution of duties as the office bearers. I derive immense honor and pride in extending all the best wishes to the Student Council. Namaskar and Jai Hind. Over to you, Tanya Beta.
Thank you, ma'am, for your uplifting and inspiring words. As we draw near to the end, I would now like to invite the newly elected President of Botanical Society, Ms. Priyanka Barua, to give a formal vote of thanks.
Thank you, Tanya. The best and beautiful thing is this world cannot be seen or even touched. They must be felt by heart. Thank you is one among them. I consider it a great privilege to propose a vote of thanks to all the dignitaries who have witnessed it as a memorable and successful event. I'm not only honored to be given this opportunity to serve as the president of the Botanical Society Deshbandhu College, but I'm also grateful to have such a talented team of council members working along with me. Today, my words are not enough to express my gratitude. On behalf of our entire botany fraternity, I would like to thank our chief guest for today's event, Professor Renu Deswal, who enlightened us with her talk. We are really grateful to her for accepting our invitation and taking out time from her busy schedule. I would also like to thank our respected principal sir, Professor Rajiv Agarwal, for always being a constant support to us. I would also like to thank our Vice Principal Sir, Professor Kamal Kumar Gupta, for always encouraging us for something new every day. My heartfelt gratitude goes to our IQAC coordinator, Dr. Aditya Saxena. Special thanks goes to our teacher in charge, Dr. Aparna Nautiyal, for being so supportive and understanding. A heartfelt thank goes to our society advisor, Dr. Sunita Malik, for all her support and advice to make this event happen. With a deep sense of appreciation, we thank our loving teachers, Dr. Sarla Gupta, Dr. Bela Bhatia, Dr. Roshni Rajmohan, Dr. Rajendra Kumar, Dr. Kumar Shantanu, Dr. Anju Chibbar, Dr. Madhurani, Ms. Anjana, Dr. Sauram Singh Dio, Dr. Rubina Chongtam, Dr. Neha Yadav, Dr. Preeti Rawat, Dr. Abhay Pratap Vishwakarma, Dr. Praveen Gupta, and Dr. Nisha Sogo. I must remark a proficient sense of gratefulness to all our teacher alumni, which we are having today in our event. One of them is Dr. Veena Ganjuma. I thank all the teachers and the students from different colleges and universities for showing their interest in this program. This event would not have been possible without your enthusiastic participation. Special thanks goes to technical and social media team, without which this event would not have been possible. A sincere thanks to all the non-teaching staffs who have always lent a helping hand. An event of this dimension cannot happen overnight. The wheels start rolling months in advance. We have been fortunate enough to backed by a team of a very motivated and dedicated volunteers. I cannot thank everyone enough for the involvement they have shown and the willingness they have expressed to complete the task beyond their comfort zone. The event is yet to complete. We have 2 inter-college competitions after this ceremony. One is Rang De Matka competition, which is a fun activity of taking trash and creating innovative pot out of it. And the another one is quiz competition, which is based on lockdown memories. I wish all the participants best of luck for their upcoming events. Once again, thank you very much all for assembling here.
So may I leave now, Sunita? You are unmute.
Sorry, ma'am. Yes, it is over now. I think Tanya just has to.
Yes, ma'am.
We have to take a picture now, ma'am. We have to take a picture together, and after
Okay.
that, ma'am, you can.
Okay. Okay, no issues.
Okay. It's not the end of the book, it's the beginning of a new chapter. With this quote, we come to an end to this glorious event. The feedback form for the event has been floated in the chat box. Kindly fill the form to receive your participation e-certificate. All the participants are now requested to turn on their cameras so that we can capture this memorable moment. Soon after this meet, we will be beginning with the events that have been mentioned in the poster. The Zoom links for the 2 events have been created and already floated in their respective WhatsApp groups. Thank you for joining this event and making this extraordinary. Have a nice evening.